way. Okay, recording is on. I'm going to give you a quick introduction. My name's Rich. If you don't know me, I get emails, Sir, Mr. Thompson, this, that, and the other. I'm Rich. I am not your school teacher, and I am proud to not be your school teacher. I am someone who's an exam coach. I'm a specialist in A-level biology, and it's my job to make A-level biology as, I'm not going to say as easy as possible, as less difficult as possible. I created the ultimate guide after years and years of tutoring. Basically, I could see stumbling blocks, be that with students who were struggling to get D's or students pushing for A stars. There's common problems throughout the course that make A-level biology really difficult, and I knew how I could solve some of those. And so I went about on a mission to create a tool that was going to make A-level biology as straightforward as possible to give it maximum impact in terms of grades. I've done basically a very exam-focused approach. So I've gone through all the past papers since 2000 and, well, the last spec and the spec before that. Basically, I've done them all. I've done them all for all the exam boards. There's probably not many people in the country who've done more biology A-level past papers than me. It's a bit sad to say, but I know all the tips and all the tricks, and I can see questions for what they mean like this, and I'm going to share some of that knowledge with you today. And I know all the exam boards, all the major ones anyway. We're going to have a little check out of that right now. So you can raise your hand. I'm going to tell you right now, I'm not going to pay any attention to raised hands. There are loads of you with raised hands right now, but I simply cannot deal with that volume of people in there. So ask me questions when I'm asking for questions, and if you're trying to ask me questions outside of that time, I've built a few videos into this to give me a little bit of time to do some, can you raise your foot? No, you can't. You can try. So I'll try and answer any questions that I can, or recurring questions. The most common ones are the ones I'm going to pick. So that's where you should be able to interact with today. Got loads of emails this week about Roy the Puma. I used to work, this is me with dreadlocks. I used to work in Bolivia with a puma called Roy. I used to be tied to him and basically take him for 20-kilometer long walks every day. So he was calm most of the time. I only have one scar that persists from him, but yeah, it was quite an experience. I just thought, your questions are great. Is that normal? I'll come back to questions over there in a minute. So yeah, I thought some of you might like to see some of Roy's to save me writing 10 emails this week, because I usually have to answer questions about Roy quite often, but he was quite a cutie when he was being friendly. Okay, so there's going to be some polls, some interactivity. So let's, without further ado, let's find out what exam board you guys are studying. For biology, obviously, please. Okay, so again, if you can make this quite nice and snappy, get your answers in super quick, then the more time I've got to give you more information. I'd like to get at least 90%, a large sample size. Yeah, if you can do this in the poll, I can see everyone pinging off in the questions. Do it in the poll. That way I can publish the results to everyone. Okay, we're at 90%, so let's close the poll. If you've not done it, you're too slow. Get faster next time. Okay, so we can see, oh, I can't see because my questions panel's in the way. Okay, so 52% AQA, so more than half of you typically do AQA. That's not unusual. And then we've got OCR, next biggest, pretty consistent this year actually. Lots of consistency amongst, yeah, that's pretty typical. So let's hide that and move on. So I want to know, does some weird things, this webinar software sometimes. Okay, so I want to know how confident you are feeling about your A-level biology revision. So we've got basically a month to go, just under. Where are you at? And again, I'm going to do it as a poll, so we can share it with everyone. Go. Okay, so again, most of you, we've just hit 90%. That's good enough for me. Let's share that. Okay, so 20% bricking it, 30% not so good, 35, third of you kind of okay. Okay, so that leaves in like the okay and under category. We've got like vast majority, 80-odd% of you in there, and 50% or more in the not so good end of the spectrum. Okay, well the good news is we can turn that around. You've got, relatively speaking, enough time to be going through most of this stuff. I don't know what's going on with my polling, but okay, so that gives me an idea of where you're at. So stay tuned to the end of the lecture. Basically, what we're going to be doing is giving away a few different copies of the Ultimate Guide. So 3 people today are going to walk out of this with a full 200 quids or 300 quids worth of Ultimate Guide package. That's awesome. If you've already bought it, then you can go in the draw for a £100 Amazon voucher. There's going to be some pretty big discounts and special offers at the end. Also, if you stick around to the end, we'll send you the recording of the webinar. There's some free downloads, and you can also obviously get Q&A, ask me some questions, and I'll deal with all that kind of stuff then. So really, really worth sticking around to the end to get all that goodness. Okay, so some of you have used this already for the free trial. It'd be nice for me to know what, how many of you have actually had some idea of using the course. How many of you know what I'm talking about, and how many of you don't? Again, so let's get over as many of you voted as possible. 80%, you're getting quicker. I like it, you're listening. 90%, let's do that and share that. So about 50-50, bang on. In fact, that is that was the referendum result, 52-48. So pretty much most of you have at least had some idea of looking at it. Those of you who don't will just see what do you guys that have used it. Obviously, I'm only expecting half of you to vote now, or to poll in this one. But what do you think about it? Like, how did you find it? I'm guessing it was a free trial that you've done. We've done loads of free trials throughout the year. The full free trial, I have to say, is finished for the year. There is no more full free trial. Okay, let's not hang around too much on this. So, vast biggest group by a long way. 5 stars, thumbs up. I like it. So yeah, basically everyone saying that it's pretty good. Makes me happy because I wrote it. Okay, so without further ado, let's crack on a little bit more. So those of you who don't know it, if you do want to go check out, there's loads of Facebook reviews. I'm going to be asking some of you that have used the course to do some Facebook reviews for me today. There's heaps and heaps out there. I'm not going to dwell on this. Just go to the Facebook, tailoredtutors.co.uk, and just find the reviews. There's heaps and heaps of them, and people say nice things, and we don't write them, obviously. So what is our mission? Our mission is to share the best possible teaching, the best possible study techniques, the maximum efficiency. So you spend the least amount of time working and the most amount of time like doing what you want to do, basically, that you're going to get more out of your grades. Whether that means you save time and you can do more part-time work and earn more cash, whether that means you can go to the park and play more football, whether that means you do more studying and get better grades in your other subjects, whatever that might be. But we maximize the efficiency. We do that by hacking the exam. We study the exams, we figure out exactly what you need to know and nothing that you don't need to know, and that's what we teach you. So, first giveaway. We set up an Instagram page last week, and so by the end of this presentation, in an hour's time, I'm going to be picking someone, who's started following our Instagram page. The Instagram page is Tailored Tutors, one word, super easy. One of you who does that today is going to be drawn at random by me at the end of the session. I'm going to get my phone out and see who's done it in the last hour. Just going to pick a name. If it's you, you win. If you've already bought the guide, you go into it, you get £100 Amazon. So pretty good. Again, there's loads and loads to do. Tailored Tutors. If you can't find it, then ask someone else. More testimonials. Let's forget testimonials there. It's awesome. And if you want to go check them out, look them on Facebook. Okay, so what we're going to do today, I'm going to start with a major content any second now. We're going to do understanding exam questions. Major issue is that students don't quite know how to understand the question, to use the right words on the exam to get the marks that you really deserve. You might revise the topic really well. You might know neurons inside out. You do a question on neurons, you write a decent answer, and you get like 1 out of 4. Like, why is that? I'm going to tackle that and show you how to understand that. I'm going to show you what to revise and how you can condense as much revision into the smallest possible space of time. It's what I specialize in. And I'm going to show you how to revise effectively so that the time that you are spending revising is exactly what you're supposed to be doing. You're not wasting any time. Okay, so what do you hate about A-level biology the most? What is it that you think is basically the most annoying things? I've picked these from experience. These tend to be the categories that people choose. If you've got some other burning ones, then let me know in the questions pane. I can see them as well. I'm going to be mostly covering some AS content. If you don't have Instagram, Emma, there will be other opportunities to win with other, yeah, there's 2 other opportunities to win the course today. So you don't have to have Instagram. Okay, and most of you have voted. Let's close the poll and share the results. So, remembering all the content, there is huge amounts in A-level biology. Most common, 73% exam technique, using the right words to get marks on the exam. Okay, so I'm going to be tackling both of those in some detail. Understanding the concepts, linking them together, third of you, motivation, revising effectively, and some of you, I mean, standard bell-shaped curve, standard distribution. Sometimes you're going to have awesome teachers, sometimes you are not. Okay. Also, this is just a quick question. How many of you got a part-time job? I'd be super interested to know. Like, I was asked that by one of my staff today, who's in Canada. Like, how many of them work part-time? And I was like, to be honest with you, I'm no longer a teenager. I don't know. So again, we're at 83%. You're getting really snappy with these. Super awesome. 87, 88. Okay, let's have a look. So, okay, 60-40, pretty much. It's 60-40. Interesting. Super good. Well, I mean, that will kind of be useful for us to know at some stage, I'm sure. I just wanted to basically answer Kate when in the office. Okay, pens out, papers out, ready. I'm going to drink my tea, and then we're going to get going. This is where it begins. Okay, so number 1, understanding exam questions better. So, command words. Super, super important. Some people call these question words. The exam boards call them command words. That's why I always use the terminology that the exam board is going to use, because obviously that makes it easier for you. This tells you how to answer the question. I'm going to ask you straight away, what is the difference between describe and explain? If that isn't immediately obvious to you, what's the difference between a describe question and explain question? You've got work to do. And I'm going to explain that to you right now. But explain means, I'm going to start over here actually. Explain means give reasons for. If you're giving me, if it's a describe question and you're telling me why this has changed, it shows you don't understand the question. The word is describe, the trend increases and then it decreases. Explain the trend. Why does it increase? What are the reasons for it increasing, and why does it decrease? So these are super, super basic. The most 2 common command words that are used in A-level biology for all the exam boards, and frequently people don't know that. So whenever I see an explain question, I scrap explain, and in my head I replace explain with give reasons for. Give reasons for this happening. Give reasons for that happening. If it's a 4-mark question, give 4 reasons why this happens. If you give reasons into the question, it automatically demystifies it a bit. It helps break it down so that it's easier to comprehend, and understanding the question is the first battle. Okay, if we're going to describe this graph, I've not put axes on it deliberately, but let's say it was going to be enzyme and temperature. Describe the graph. Graph increases from 0, peaks at, quote, a point of interest, or the peak is typically the point of interest, and then decreases rapidly to 0. So that's going to be my description. I don't have to give reasons. In fact, if I'm giving reasons, I'm demonstrating that I'm not understanding properly. Explain the graph. Well, increase in temperature, increase in kinetic energy, more collisions, more enzyme substrate complexes formed. So that is the reasons why the graph is increasing. Obviously, every graph is going to be slightly different, but understanding the principle between describe and explain, essential. Moving on down the page, we've got name, state, give, calculate, define. They are going to be 1-mark questions, or possibly calculate a few more questions, but there's no mystery to the question. So I'm not going to take much more time on that. You know what you need to do. You either know the answer or you don't know the answer. Either way, there's nothing you can do. There's no question analysis you can do there. The other 2 main ones that are on the page are suggest. Suggest are typically the most hated. Okay, so what can you do to help you understand the suggest questions on the paper? You are going to have to stop a moment, think, what is this similar to? It's going to expect you to apply your biological knowledge to a scenario that you've never come across before. Maybe it's about a weird neuraminidase enzyme that you've never heard of, or maybe it's not even an enzyme. Maybe it's a totally different type of protein, but it's a protein. What do we know about proteins? We know it has a primary structure and a tertiary structure. Its shape is very important to its function, so we can apply what we know in biological knowledge into another region. So it does going to take you a little bit of time to stop and think about. Also, a bit of common sense. Some of them are about like the seasons. How would you expect this to change over 12 months? And maybe the country is going to be the UK. Well, we've got 4 seasons. So they're going to expect you to do a little bit of brain work thinking about the question here. It's generally common sense. Don't overcomplicate it and put in as many key terms into your answer as possible. It goes for all your questions basically, but especially suggest. Last one I'm going to tackle in any great detail here is evaluate. Evaluate questions are all about giving the advantages and the disadvantages, or the pros and the cons, the reasons for the supporting the statement, the reasons against supporting the statement. So maybe the data, data is actually plural, these data do agree with the scientist's conclusion because there's a large sample size, it was conducted by scientists who are not biased. However, so we've given 2 reasons for, however, there was no control used in the experiment and all the patients were healthy and this drug was supposed to be testing it, or how we don't know how it will perform on unhealthy patients. Something like that. You've always got to give me both sides of the question. Do I recommend giving an conclusion to evaluate questions? If it's 4 marks, most of them are 4 marks, then there's no marks for a conclusion. If it says, if it's sometimes the question very, very rarely will say and conclude like what, give the arguments for and against, basically evaluate this and then come to a conclusion. If it expressly tells you to give a conclusion, then a 1-sentence conclusion is all you need. But generally no. For evaluate questions, reasons for, reasons against. Okay, so next up, after doing all the past papers basically available in the world ever, these are my 9 top tips to how to analyze the question, to make sure the question is not tripping you up. And just a little point, number 7 here is when we begin writing. Most of this is done in your head because you're going to have to spend more time thinking about the question and analyzing the question than you do at the moment, almost certainly. Okay, so 1, 2, 3. Read the question. What's the command word? Easy. How many marks is the question worth? Very important. That shapes the level of detail you're going to give. It shapes the way you're going to answer the question. If it's 3 marks and it's explain how this would affect these 2 things, is a bit tangential. But if you've got 2 things in 3 marks, then one of them is probably going to be affected and one of them is probably going to be unaffected. And so which ones, where is it more important to put your answer, the weight of your answer? And the number of marks indicate where our answer should be going a little bit. It's a clue. Third, we want to identify the topic of the question. Now, this is not always straightforward, and quite often the question is setting you up with an introduction. The great one I saw the other day, classic example, all about photosynthesis. Plants here, photosynthesis that, temperatures this. Okay, explain how an increase in temperature would affect the rate of photosynthesis. Now that question, despite the fact you've seen a graph and you've read the blurb, it's all about photosynthesis, that question is not a photosynthesis question. That is an enzymes question. Explain, give reasons for how photosynthesis, rate of photosynthesis increases with temperature. Well, we need to know that photosynthesis is controlled by enzymes, but hey presto, basically all biological systems are. Then we need to know, that's the only thing we need to know there. Then we can say increase in temperature, increase in kinetic energy, more enzyme substrate complexes. That is an enzymes question. Just because the framework of the question is about photosynthesis is irrelevant. And you've got to be able to get to the point you can see past that confusion and like, what is the question on about? And we're going to do some practices of that. We're going to be doing some mostly AS content today. Again, I'm showing you the tools that you need. This applies to AS questions as much as it applies to A2 questions. So you're going to be getting great value out of this, basically. Next up, you're going to rephrase the questions and make the questions easier for you to understand. So if it's an enzymes question, get rid of the nonsense about photosynthesis and phrase it as an enzyme question. So you can get the question number 5 100% clear in your mind. You need the question clear in your mind. I work with students one-on-one all the time, and frequently a student will answer a question, 4-mark question, and I'll take the answer in the paper away. It's like, okay, what was the question? And they're like, was something about you've just answered it. If you don't know what the question is and you've just answered it, that's not going to be a great answer. You need every single sentence of your answer needs to relate to the question. So you are going to be getting better and better at this. This is in the handouts. This question analysis will be emailed to you after the webinar. Use this with a past paper. Work your way through past papers. It will take you a bit more time, but do this for every question, and you'll start to see the patterns of how the answers build up. Next up, we've still not started writing yet. Next up, this is all going through in our heads. We are going to brainstorm all the relevant key terms on that topic. We've got the topic and the question clear in our mind. What are all the relevant key terms? And like one will trigger another one, and that will trigger another one. Maybe it's nerves. We've got sodium potassium pump. We've got resting potential. We've got generator potential. We've got depolarization. Boom, 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 and it'll be like a little cascade, like a dominoes, of one key term triggering another key term, and like putting that framework of key terms in your head is really important, because then we get to number 7. We're going to start writing. And when you start writing your answers, you've all seen the mark schemes, I hope. If you've not seen a mark scheme, shame on you. Make sure you're downloading all the past papers and mark schemes. You will see there's only about 5 words per mark on the mark scheme. It's not looking for huge volumes. It's looking for very focused, single short sentences that directly relate to answering the question. Each sentence, number 8, must have at least one key term in it. So you're going to think in bullet points, write in full sentences using a full stop as a bullet point. 7 to 10 words max. If you're writing long sentences and if you're using all the text space available to answer your questions, I'm going to challenge you to go back and write only maximum half the amount of lines. Try and answer the questions in half the amount of lines you have available in practice. Force yourself to think what is important. Half of what you're writing, if you're writing in all cover, filling the whole space, is irrelevant and not going to get you marks. So only write those key term focused sentences that are going to directly relate to the exam question. And then at the end, you can reread your question and add in any key terms that you've missed. Won't you run out of time? Okay, so Moshe, in practice, you're going to have to practice this. This will be slow. In past papers, you're going to have to go through this. Now, this is automatic for me. I read through the question, I've picked the command word, how many marks it is, and I've rephrased the question when I've read it twice. By the time it takes me to read the question twice, I've done stages 1 to 5. Then I have a quick brainstorm of all the key terms that are relevant. That's going to take me another few seconds. And then I spend probably as much time analyzing the question, because when you're not writing junk anymore and you're only writing pure gold for answers, I can write on a 5-mark question, I can write 5 sentences and get 5 marks in no time at all. So in practice, this will be slow. The more times you use this and apply it, the faster it's going to get. Stages 1 to 5 should become second nature. 6 is going to require a little bit of thinking time, and then 7, you're writing your answer. So it's not going to delay you too much if you practice it. Okay. So now we're going to look at a real exam question. Maltose is hydrolyzed by the enzyme maltase. Explain or give reasons, remember, why maltase catalyzes only this reaction. Now I'm going to ask you in the question comments here, what is the topic of the question? In fact, let's just go back. We're going to look at the command word, how many marks, and what's the topic of the question. Okay, be as specific as you can with this. I'm seeing enzymes. Everyone's saying enzymes. One person saying carbohydrates. Complementary active sites, enzymes. Okay, a few of you are getting a bit more specific. Enzymes is too big a topic, I think. Can we get into enzymes, structure of enzymes, specificity of enzymes? Okay, induced fit enzyme substrate complexes. Okay, so we're getting much better. We're going from enzymes and carbohydrates to, I'd say the topic of this question is, what have we got to do? We've got to give reasons why maltase only catalyzes one reaction. So what's that about? Why do enzymes only catalyze one reaction? It's asking us, why are enzymes specific? Enzyme specificity. Nailed it. Okay, so this is a video from the Ultimate Guide that basically catches there's hundreds of questions, literally hundreds of questions on proteins of changing shape and not being complementary to the thing that they were complementary to before. This video from the Ultimate Guide is like the catch-all answer to all of those questions. They're just a reskinning of the same question. So I'm just going to stop my video, or like my head video, for a second, because it's going to make your video a bit bigger. Okay, so I'm going to try and tackle some of your questions now whilst this is playing.
Continuing with enzymes. Okay, so enzymes, there is a classic question that comes up all the time. In fact, it's more of a protein question. It's most commonly asked as an enzyme question, so I call it the protein shape question. Now, this could be enzymes, which it most commonly is, but it could also be antibodies. Remember, we've got EATS, enzymes, antibodies, transport, and structural proteins. They're the different types of proteins. So this is going to be the, now the key terms are going to go in red, tertiary structure of the, now what's the important bit within a protein or an enzyme? It's the active site, or it would be the variable region of an antibody, is complementary to the shape of the substrate and forms something that needs to be in all of your answers when you're talking about any of these things, an enzyme substrate complex. Quite commonly, this is written as an ES complex. This could also be an antibody-antigen complex, depending on the type of molecule. Now, this could be, this is for a functional, obviously this forms an enzyme substrate complex, so it's working, it is functional. What about, you get a question that's asking you why a protein is no longer functional? A non-functional protein. Again, this could easily be an antibody. Now, there's two reasons why this might be non-functional. The most common is going to be a mutation. Now, if it's a mutation, we're going to look at this in a lot more detail when we do the science behind it. But this changes the primary structure. So that's the sequence of amino acids. And remember, we can write primary as one like this. The primary structure of the protein, and if you change the primary structure, this changes the tertiary structure of the protein. And now, actually, the rest of this is going to be the same as our answer from over here. So, if the enzyme is denatured, remember that's the temperature causes the molecules to vibrate so much it changes the internal bonding. So this changes the ionic bonds, which changes the tertiary structure. Now, both of these things, we've now changed the tertiary structure, so now we can continue both in the same. This changes the shape of the active site. Now we can see a pattern emerging. We've changed the shape of the active site. The substrate, excuse my handwriting, the substrate is not complementary anymore. And then finally, what do we need to say? We always need to mention the enzyme substrate complex. So, no enzyme substrate complex. And that's why it's non-functional. So you need to think with these questions. First of all, is it functional or not functional? The answer has got very much the same structure. First of all, think, is the primary structure relevant? Has the sequence of amino acids changed, yes or no? If the sequence of amino acids has changed, then you need to mention the primary structure. That's generally a result of a mutation. The change in the structure of the primary structure is going to cause a change in the tertiary structure. So we can write tertiary like this. We can also write tertiary with 3'. And then we need to say, is the substrate complementary to the active site? So we always have to mention the active site, and we always need to say if it's complementary or not. And then we need to say, finally, does it or does it not form an enzyme substrate complex or an antigen-antibody complex or antibody-antigen complex? If you can answer the questions using that terminology in that structure, you will get full marks on all those enzyme questions.
Let's turn my video back on. Last time I forgot to turn my head back on. If I'm doing anything stupid and I'm not done anything like that, then do let me know. Oh, not that it wants to. Okay, perfect. Come on, computer. It doesn't want to turn my camera on. Well, you're going to have to make do with my voice for the time being. Okay, so I could always turn on my other camera. We'll try that, shall we? Try this one over here. I don't know what. Hello from a different angle. Okay, so yeah, my computer over here is just having a bit of a freak out at the moment, but that's where the presentation is. So, oh, it's blue screen of death over here. Okay, better. Okay, so moving on. He says, with a jammed computer. Okay, I'll just field a few questions whilst everything comes. Can't see me. Are all of my videos on YouTube? No, they are not. A couple of them are. You can see me. I think you can. You'll see. I'm looking at this camera now as opposed to the one over here. But my laptop over there is just having a bit of a minor freak out. Okay, you can all see me. I think it's just a few of you. So a couple of questions. A lot of you are asking me, what's the price of the guide? Well, we're going to come to that at the end. I'm going to give you some extra special, super amazing discounts. It says I'm frozen. Some people say that they can't see me, so there's definitely something freaky going on. Let's quit this, I think. Not the best time for your computer to go down, is it really, when you've got 300 people just kind of listening to all sorts of things? Okay, let's. Appears to be happy again. Let's try this camera and see if that will turn me off. Oh, look, you get to see me twice. Hello. Let's turn this one off. There's certainly one mustache is enough at any one time, I think. Okay, so here we go. We are back in the room. Okay, so just reminding ourselves of this. This will be in the email, in the handout. Following up the session. Use this in your past papers. It's really going to make a big difference. This camera is much better. Yeah, this is a proper camera. I don't know why I think my Mac's just a bit hot. It's pretty warm in here today. Okay, so going back to this question, how do we analyze the question to make it easier to understand? Maltose is hydrolyzed by the enzyme maltase. Explain why maltase catalyzes only this reaction. Well, for me, straight away, get rid of explain, now give reasons. 3 marks, okay? So this is not about maltose particularly. This is about enzyme specificity. So my rewording of the question is using maltase as an example, give reasons why enzymes are specific. Now, let me know which question's easier. Is it their question or is it my question? Which one's easier to answer? Tom, big up the Ashcombe School. I have just been to the Ashcombe School. Ashcombe are one of the schools that use the course that and I guess, Tom, you are one of them. I'm actually speaking to you very close to the Ashcombe School, just around the corner, actually. My question. Okay, so using those 9 bullet point steps, do this in practice as much as you can. It will be slow to begin with, but you are going to be able to get to the point where you can demystify these questions. Okay, next step was have a quick brainstorm. Boom, active site, enzyme substrate complex, protein, boom, 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 complementary. You can, at this point, as long as you're on topic, that brainstorm of key terms is really going to help you sort of grab all the information that's relevant to the answer. And now you're going to put them in order, a logical order, short sentence, minimum 1 key term per sentence, and we're going to end up with an answer something like this. The active site of maltase has a specific tertiary structure. Only maltose, or the substrate, is complementary to the active site, and only maltose can form an enzyme substrate complex. 3 standalone single sentences that each relate to the question in the correct order. So no, you don't have to write the brainstorm. The brainstorm is purely, in an exam, you're not going to have time to brainstorm them out. If you want to do that in practice and write a little brainstorm, it's good exercise, but in your exam, the reality of the situation is it's going to be in your head, boom, 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 boom. Okay, and then we're going to come up with, so just looking at the screenshot, tertiary structure of the active site is complementary to the shape of the substrate, forms an enzyme substrate complex. Right, this answer answers all of those questions. You just need to see the question. Okay, so now I want to know, I've been ignoring you, doing lots of yapping, what is your general revision problems. What do you hate most about revision? Okay, so okay, if there's any other options, you're welcome to post the other options through to the comments. I'll come and check them out over here. Okay, most of you voted, 90%. Let's have a look. Motivation, boredom, too much to do and don't know where to start, and then a bit of procrastination. So they're pretty big topics in there. Okay, so distracted easily by the smallest thing. So that's kind of, yeah, okay. All of that's coming up in, has got some great tips for how to avoid that kind of stuff. Okay, another giveaway. Someone else is going to win. I should check Instagram, see how many of you have been liking my page. So the Instagram one's still running. That will be, the winner will be drawn at the end of the session. Another tip. Okay, so I want a revision tip. This is going to be A-level biology, or it's got to be general revision for any subject. But what's your advice to other A-level biology students right there, right now? We're going to make these into more Instagram posts. Basically, we're going to pick the good ones. That's why it's a good idea to follow the page, because we're going to be making some of your ideas real Instagram posts that are going out to the masses. Basically, you've got to email them to me. That's the only place we're dealing. People sent me messages on Instagram yesterday. I only can pick them from one place. So email me your revision tip, and I'm going to pick a winner straight away afterwards. I'm going to, within an hour of the end of the show, I'm going to pick a winner by, what time do we finish? 3:30. By 4:30, I'll have picked a winner and emailed them. I'll also post the winners to the Facebook page. So if you're not sure whether you've won or not, then you will find out there. And if you do end up buying the course and you do win, we'll just refund you, so you don't end up out of pocket. But send me a revision tip that we can make into a Facebook post or an Instagram post, and that's basically what we want you to do. All entries are going to count as long as they're half relevant. If it's total nonsense, then we'll just then ignore you, but we pick you at random. You've got maybe half an hour at the end of the show, so scribble it down, take a photo, you can give it, type it out, however you want to do it. If you want to make it look beautiful and send it to us, then we might just post it straight up as it is. Okay, so how to tackle questions. Next up, we're going to look at what to revise. So a lot of you said that there's too much, you don't know what to do, and a lot of you have been asking me about past papers. So there are lots of things that we can do to modify the way we revise to maximize this efficiency. I'm going to suggest in the follow-up email that you are going to get, you don't have to ask me where it is, it will be sent to you within the hour to the email address that you enrolled in the webinar with. You're going to get a link to my Dropbox account with my entire stash of AQA papers, OCR papers, WJC papers, and EdXL papers. So all the old specs, all the new specs, all the very old specs, there's going to be heaps and heaps of papers there. So you can download them all, and you can search through those papers and find questions that are on the topics that you want to revise. So maybe you want to revise, they are all free. Yes, they are all free. I don't charge people for things. They're all available online. They're just difficult to, it just takes hours to download them, rename them, put them in organized folders. I've done all that for you. You can digitally search for the key terms, and you can then choose transcription, you can choose neurons, you can turn epistasis, whatever the topic you want to revise is, and you can search those documents and you can find the questions on those. The old past papers are about 75% the same. There is a slightly different emphasis on maths. There is some content that is gone. There is some content that is new. Okay, know your specification. If you know your specification, then if you get a question on something that you've never ever studied, then clearly it's just something that you don't do anymore, and therefore you don't have to worry about that. So don't let the old past papers freak you out, but that you are the guinea pigs, guys. You've got like no past papers to work with. Past papers are so important for training yourself how to understand the questions and how to answer them in the right words, that you're going to have to use some of the old past papers. Rich, you're the best. Thank you so much for this. Thank you, Olivia. So you can basically look at the questions, and you get to see the common answers as well. You can also see when you're searching these papers, how often do they ask the question? If you search for a key term and you see that it's in one past paper in 2004, is it that important? No, not really. But if you do a search for a key term and it's in like every year or every other year, then maybe that's a good topic to be revising because it comes up all the time. So you can also see whether you search for a key term, is it on the mark scheme? Is it on the question paper? If it's in 10 mark schemes on one question paper, then you know that you have to use those words in the marks and in your answers. Okay, so another video just coming up here. I'm just going to kill my video again. Hopefully it doesn't freak out on me again like it did last time. And this is going to take you through some of those tips. Hey guys, so you've just seen my video lesson on enzymes and the key terms that you've got to use in the exam to get those great marks. I'm going to show you a little inside sneaky secret as to how I built the course and how I know what those key terms are going to be. These are all the exam boards out there, so well, all the common ones anyway. I'm going to use, for this particular example, AQA, just to show I've got all the exam board papers ever. So I've done lots of exams, lots of times. I'm going to use Unit 1 for this example because it's the most similar to the course that you're doing now. Let's take tertiary. Tertiary structure. Okay, wow, look at that. So MS is mark scheme, mark scheme, mark scheme, mark scheme, mark scheme, all mark schemes. That tells me immediately tertiary is a word that is essential to use on the answers of your questions because it's a key term that will get you marks. It's not on any of the question papers, which means, well, they want you to use it. So this is how I can build a list of what they want you to know, and I know what they don't want you to know. So it helps me write my courses so that they're perfectly tailored to the exam boards. I can hear you asking, what about not AQA? Well, there's a couple of AQA examples here. I've also got loads of other exam boards open as well. Let's look at this question. Scientists use the antibody to detect an antigen on the bacterium that causes stomach ulcers. Explain why the antibody will only detect this antigen. Well, what's this question saying? Give reasons for that. It's exactly the same as the question we looked at the beginning of this webinar, and that's why is a protein specific? It's a different protein, but does that matter? No, not really, because the answer is the antibody has the variable region instead of the active site. Specific primary structure, the shape of the tertiary structure is complementary to the antigen, forms a complex between the antigen and the antibody. Well, this is the freeze frame from the end of that lecture slide. Here we've got the tertiary structure, variable region is complementary, antigen-antibody complex. Lovely. Next one, okay, it's a graph question, totally different way of asking the question. Give reasons for the shape of the curve between 30 degrees and 50 degrees, okay? Boom, it's denaturing. That's what's going on there. So, enzyme denatured, hydrogen bonds changing, tertiary structure change in the shape of the active site, the substrate of the protein no longer fits, does not form an enzyme substrate complex, and the last one is that the more enzymes are denatured, the higher the temperature gets. So that's, but you can still get 3 marks here without even thinking about the graph, really. It's just pure theory. How does the notes relate to this? Well, denatured, change to the bonding, change the tertiary structure, change to the active site, not complementary, no enzyme substrate complexes. Does it get 3 marks without even thinking about the question? Yes. OCR now. Suggest how changing the sequence of DNA nucleotides could affect the final product that DNA codes for. Bing, first thing that goes off in my head when I see this, the final product that DNA codes for. Why haven't they said protein or polypeptide here? Because they want you to use it in the answer. That's why. If you ever get a question where they bend over backwards to not use something that should be in the question, that will get you a mark in the answer. Okay, so let's see if OCR are any different. Different sequence of amino acids or different primary structure. Different protein, or the protein folds up differently, or there's a different tertiary structure, no longer functions, or different function, and except a super example, e.g. active site of the enzyme no longer complementary to the substrate. So again, change the primary structure, change the tertiary structure, change the active site, not complementary, no enzyme substrate complexes formed. At Excel, I'm not even going to show you the question, this video has probably made the point well enough already, but at Excel, just to show you, enzyme is a protein, reference to the tertiary structure, reference to the name bonds, e.g. ionic hydrogen bonds, between the R groups, generally where the big bonding takes place, reference to the active site, idea of specificity of the active site, like the questions we've looked at already. Last but not least, WJEC, again, answer only, enzyme is denatured or changes the tertiary structure. Breaking of the hydrogen bonds, active site is changed or active site is denatured, substrate cannot bind, less enzyme substrate complex is formed. I hope that I can show you that using these mark schemes are absolutely essential. I know what you're telling me now is that you don't have enough time, and you're right, basically you don't have enough time. It took me 3 months full time to even begin to get my head around
all those key terms that had a few different examples to deal with, but it takes a long time because what you've got to do is open up a question like this. You can then do your search for tertiary, and then you can find the question where tertiary is. This is the one here, so it's 5C, and you have to go back to the question paper and then get rid of this search term, try and find that exam board. It takes a long time, basically. You don't really have that time. Exam is next week. Basically, you can use this tips as much as you can. The course is all in one place. The Ultimate Guide to AS Biology is super condensed. Read what people have said about it. I'm not the only one to believe it's totally awesome. It works. Loads of schools are using it, and all the heads of sciences that I've spoken to say their kids love it. So try it. There's a 100% money back guarantee. I can't tell you enough how much it's going to hit.
Okay, let's see if my camera's going to turn on again. It's going to do a little bit of freaking out. Okay, so whilst we're waiting for that, yeah, perfect. Okay, so let's move on. You can see me again, hurrah! Lots of people asking questions. Do I do individual tutoring? I do, but A, I'm very expensive, and B, I'm fully booked. There's absolutely no, I'm working like all hours of the day at the moment, and there's just not enough time. If you do half as many hours at your desk as I'm doing in a minute, you'll all get A stars. Yeah, I've been finishing at like 11, 12 at night every night, getting up early, working all day. So I do tutoring. If you're an AS student and for next year, then maybe it's an option. You don't want to know how expensive I am, to be honest with you. I'm not even going to tell you. Let's just say the course is very good value for money. I just got offered, okay, I'll let you on a sneaky secret. I just got an email offer to do GCSE tutoring for a student, online GCSE student. They're doing double science, and their parents want them to do triple science to be strict, so they want me to teach them the extra stuff that they're not going to do at their school. They offered me £180 an hour a week for 12 months, and I said no. That gives you an idea. Okay, so grade boundaries. I'm going to do most of the questions at the end. Okay, so someone's saying if they complete the course, you're going to get an A star. Obviously, I don't know how much work. If you complete the course, we guarantee you're going to go up a grade. Whatever grade you're at when you start, you fill in a form, let us know that. If you complete the course and you don't go up a grade, we give you your money back. So that's how much we know. Obviously, some people are going to complete the course and get A stars. Some people are going to complete the course and go from a D to a B. Some people are going to go to an A and A star. So it totally depends on the person. I can't, without knowing you individually or having tutored you, I've got no idea how you're going to do. This is another little sneaky inside secret, how to write. You've got a small amount of time left. How to write those like summary notes of what the exam board want you to do from in the fewest possible words, trying to shape your notes to match the exam mark schemes as closely as possible. So let's check this out. So I'm going to share with you an inside secret and show you how I've built the Ultimate Guide to AS Biology. It took me months to go through all the past papers of all the exam boards that exist. I've gone through the very old specs, and the new specifications that are out there. This is just an example of how you can use those downloaded past papers and mark schemes to write super condensed summary notes, like the ones I've put in the videos. So here we've got, these are AQA papers, it doesn't really matter, the theme is the same. Some substances can cross the surface cell membrane of a cell by simple diffusion through the phospholipid bilayer. Okay, describe other ways which substances cross this membrane. Well, we've looked at finding the command word describe. It's 5 marks. How can we rephrase this to make it easier? So it's basically describe in full detail, because we've got 5 marks, all the ways that substances can enter a cell, but not by including simple diffusion. So if we look at the mark scheme here, we've got osmosis, so we've got water molecules from high water potential to low water potential. You can say through aquaporins or water channels. You don't have to get all of these. You need to get a total of 5. You may never heard of them. They're not very common. By facilitated diffusion, you don't get a mark for saying that. It's unusual that they don't credit the actual methods, but it's saying how. So the channel protein or carrier proteins, down the concentration gradient, it's still diffusion. Active transport, you don't get a mark for that either, but through a carrier protein or a protein pump against the concentration gradient using ATP. Every single time you mention active transport, say it's against the concentration gradient using ATP. Every single time. It's always marks for all the mark schemes. Phagocytosis or endocytosis or exocytosis, it's saying into the cell, so we can't say exocytosis. Engulfed fusing membranes or fusing vacuoles or vesicles Exocytosis is the vesicles fusing and going out. I think this question actually said going in, or did it just say crossing? And cross the cell surface. It didn't give a direction. Okay, simple. Many substances enter and leave the cell by crossing its cell surface membrane. Describe how substances can cross the cell surface membrane. These are real papers. They're not specimen papers. These were both SAT. They're both from the same course, from the same exam board. So if you'd done the June 2010 paper, and then you'd done the January 13 paper for real, you would have got 5 marks absolutely free, because look at the mark scheme. Simple diffusion from high to low concentration gradient. Small non-polar lipid-soluble molecules go through the phospholipid bilayer. You can say large or polar molecules go through proteins, carrier proteins or protein channels. Water molecules move by osmosis. High water potential, low water potential, active transport from high to low concentration, from low to high concentration against the concentration gradient using ATP. In fact, that mark is, for some reason, they've separated it, it's down there. Facilitated diffusion involves proteins, carriers, or protein channels. And then you could reference the glucose sodium co-transporter, or they can reference endo or exocytosis here. So the mark scheme is basically exactly the same, and you can use these to summarize what in your textbook will be 5 or 10 pages. And look at it, it's in 10 sentences on this page, and these are the things they ask over and over again. I cannot overemphasize how useful these long answer questions in the previous past papers are for writing condensed summary notes. They show you exactly what your exam will want you to know. So do it. Okay, so loads of nice comments going in there. I left my video on that time, never mind. You can see me like writing my comments over there. Okay, so let's not look at that. That's all too boring. Okay, so you guys, I feel sorry for you guys. You guys are the guinea pigs. How are we doing for time? Whoop, it's 55. Okay, so it's super tough. You've got the new spec. Teachers don't know the course very well. There's massive volumes. There's lots of specific key terms, loads of questions that are tricky examiner questions that you don't quite know how to understand. I know all of these problems. It's my job to know these problems, and I try and solve them as best as I possibly can. Compared to the textbooks, I'd love those of you who know what the textbooks are like. What are the problems with the textbooks? Let me know what your problems are with the textbooks in the questions over there, because like I know they're too much waffle. They don't they just don't tackle the exam. They don't prepare you for the exam very well. So I try to basically make you write your own textbook. That's what my course does. You basically create your own textbook in your own handwritten notes. Okay, so third giveaway. I'm going to give another one. And some of you obviously have been here before because you're asking me about leaving Facebook reviews. If you have used the course, only people have used the course. Don't go making anything up. If you leave a Facebook review yesterday, if you're super keen to win the course, this is the one to do, because the fewest people can be bothered to do this typically. Yesterday, I think there were only like 17 people. There were 1 in 17 chance, or maybe it was even less than that. It might have been even 12 people, 1 in 10 chance of winning the ultimate guide. So if you leave a text review, it has to be public. I'll pick it from the ones that a lot of people have said they left reviews, and they've shown me a screenshot. They have left a review, but they've said it so that only them and their friends can see it. That's no use to me, really, to be honest with you. You need to be able to leave a review that's viewable to the public. So just make sure the privacy setting when you leave your review, whatever you type, is set to public. I'll be picking the ones that are publicly displayed. I'm sorry, them's the rules. So like the page, leave a Facebook review. Anyone who does both of those things, we pick one at random, and I'm going to post that basically by 5 o'clock. As soon as I'm done here, I'm going to pick an Instagram winner before the end of this one. I'm going to do the revision tip. I'm going to do the Facebook review. If you have used it, then I'm going to pick those winners as soon as I'm done here, basically. Okay, so that's probably enough time spent there. Okay, so third section, how to revise effectively. This is going to get rid of all your sort of procrastination problems, your motivation problems. It's going to tell you what you should be revising and how you can do it most efficiently. This is my email address. It's there on the page. Oh, rich@tailoredtutors.co.uk. I thought it was on that page. It's not. So rich@tailoredtutors.co.uk. I'm sure it's on the website tailoredtutors.co.uk. You'll find it. It's not too tricky. Okay, so FAST. A load of neuroscientists, a bit of educational psychologists, teachers with more than 100 years worth of teaching experience combined, plus I shaped this basically, plus my ideas, have basically developed this learning system for you to use, especially when you're revising. But we teach a lot of good technique, good productivity. So fast, focus, associate, repeat, summarize, and then teaching and testing. You apply these principles. I'm just going to get you to do this for one day of study leave at some point, or one session, even 1 hour, and you're going to see the difference this makes to the amount that you learn and the amount of time that you have to do it. So F, what does F stand for? I want to see it before I move on. Focus, perfect, Georgina. Emma, you're in there. Yeah, Wazil, Abby, yeah, you're all nailing it. Okay, so focus. What do I mean by focus? Okay, so focus means that when you sit down to revise, to work, to get your knowledge in your head, basically you look at only one thing. Right now, if I look over here, we're going to see this, but you've got the camera of me and presentation of me. I did a lot of research in terms of learning theory when I created my course, and I was going to have this format. I was going to have me talking like this, and I was going to have the notes coming up elsewhere. And if you split people's attention between two different places, their ability to recall that information, they're divided. Your brain is trying to process two things at once, and it can't do it as well as it is for giving all your focus onto one thing. So that's what this is about. This is about basically exam conditions, maybe some instrumental music or some chill music in the background, no phone, no Facebook, no distractions, no social media. Basically, the biggest, most important thing on here is this one. Write down what you are going to do during that session. I like to work for an hour. It gives me then 20 minutes for a break afterwards, and I can actually do something half useful in that break. If you work for 20 minutes and then you have a 10-minute break, it tends to be a 20-minute break. It tends to mean that you don't get very much done. I like to do 1-hour desk sessions. And you have to write it down. Knowing what you're going to do is not enough. You physically form a contract with yourself. I am going to, what am I going to do? I'm going to watch 4 Ultimate Guide videos and take notes. I'm going to do past paper questions for 45 minutes and mark them for 15 minutes. I am going to summarize protein synthesis in an A3 poster. Right? Whatever your topic, what your target is for that hour, it has to be a single target that is actionable in that hour. You can't just write revise maths, revise biology. That doesn't work. You've got to give yourself a specific goal that you can do in that hour. Sit down and only do that one thing. What I like to do in the morning is I like to have a cup of tea, a cup of coffee. If it's a Saturday, Sunday, a study leave day, then I can plan, okay, biology, I've got maybe 3 sessions I want to do for biology. What am I going to do during each of my 3 sessions? Then when I come down to sit down and do biology revision, I can pick one of the 3 things off my list. Oh, I'm going to do these past paper questions. And you sit down, you set a timer, bang, you go and do your hour. Really, really important. How many hours a day? Totally depends on if you're going to college, whether you're not at college, how many subjects you're doing. I would aiming to be trying to do almost 1 past paper a day and at least an hour of some other stuff. Okay, so A is for associate. Okay, so making associations between things makes your brain give it 3-dimensional context. It puts it into an understanding, linking it to your brain. I call it a memory hook in the teaching that I use. So like meiosis sounds like mayonnaise. Mayonnaise looks like sperm. Meiosis is cell division to form gametes. Okay, so this is basically the easiest way of linking these together. It doesn't have to be that smart, doesn't have to be that funny, it has to make sense to you. An association is better than no association. It's amazing how powerful that is in order to dredge information back when you're sitting in the exam. You don't have your notes, you don't have the textbook, so you're going to have to do it from what's in here. And by putting a bit more effort in and linking things together and making associations in your notes, it gives it 3D context that, again, makes it so tangible and easier to remember. R is for Hassan, straight in there on the B of the bang. On the R of the R, it is repeats. Okay, so sadly, the way the brain works, we learn through experience, we learn by doing repetitions, we get things wrong, we then get it right again, so repeat, repeat, repeat. You're only going to know it if you actually do a few repeats of it. So I've designed the Ultimate Guide to do this as little as possible, but you are going to do some repeats. No one learns anything perfectly the first time. Not even me, sadly. S is for summarize. And again, Hedwin, Wazil, Sylvania, all in there straight away. Okay, so summarize. Writing summaries is basically going to test your ability to condense down. Let's say you've read a page of the textbook. Well, I can tell you that 75% of that is not going to be in the exam because I know what's going to be in the exams. And you've got to basically extract that important information and write short. I like bullet points. Bullet points are going to, because it matches the mark schemes of the exam. That is the best thing that you can be doing, is trying to prepare yourself for those short single sentences, single key terms, and write short summaries. Describe the process of synaptic transmission in 10 bullet points. If you can't do that and there's holes in your summary, then you can go back and you can fill in your holes. It doesn't have to be total knowledge, but testing yourself on that, which is the last one, teaching and testing, is basically a great way of solidifying your knowledge. Try teaching your mum or your dad. They're going to be blown away. They have no idea. Unless they're a proper biologist, they're not going to be able to follow any of what you're telling them, but show off. Go and teach them. Teach your brothers, your sisters, your classmates. Teach your cat, your dog, a dictaphone. Like, verbalizing it is so, so, so important, because verbalizing it, again, it gives it it gives it another dimension. It helps put it into structure. If you're trying to give a verbal summary in 5 minutes of a process and you come up with stumbling blocks, then you're going to realize the bits you do know. You're going to realize the bits that you don't know. Also very important with past papers. Testing yourself using past papers. If you're not getting full marks, that's okay in past papers. Nobody gets full marks anyway. But the bits that you know already, then prioritize your revision on the bits you don't know. What are the questions you got wrong? I'm going to generalize a bit here. Typically, girls are going to be, they don't want to do past papers until they've finished their revision, until they know everything. And that is the wrong approach. You should be using past papers. If you can do a past paper and you get 50% right now, what are the topics you got 50% correct on? Well, they're kind of in there already. You should be spending the rest of your time on the bits that you did 50% that you don't know. People love revising the stuff they know because it makes them feel good. Wrong technique. Revise the stuff you don't know and the core concepts that come up over and over again. You can only teach someone when you know it well. So by putting you in that situation, it's going to show you what you know and what you don't know. I've actually changed the video in my follow-up email. It's not the one from David Myers. If you want it on Facebook, on YouTube, it's available. There's another video on the testing effect, which I think is even better. It will be emailed to you afterwards. Yeah, I'm not going to answer that question. Okay, so boom, boom, boom. 3 major topics done. We're an hour in. We've got some time for questions coming up. Okay, so just as a little bit of a recap. Email address again is rich@tailoredtutors.co.uk. Okay, so the Tailored Tutors mission. Again, we're just basically trying to make your lives as easy as possible. It's our job, so we make it a full-time job for a few staff to basically figure out all the best possible ways of teaching and representing the data, making the best possible videos, condensing all the stuff that you need to know, stripping out all the stuff that you don't need to know, and giving it to you in the smallest, best possible package ever. That's basically what we do. We try and help you hack the exam. How to understand exam questions better. The email that you're going to get afterwards is going to give you that 9-point list of how to analyze questions that's going to train you how to figure out and demystify questions. Knowing what to revise. Basically, I'd recommend using my course. Of course I would, but it's not possible for everyone. So use the past papers that are in the follow-up email. You have to laborious. It takes a long time searching for those key terms and going through them and finding the questions. You can do it. I spent 2 years of my life doing it and organizing massive documents of compiled past papers. That's how I know how difficult it is, but you can do it. And how to revise effectively, fast learning. Try it for 1 session, no phone, exam conditions, write yourself a target of what you're going to do, and do that one thing only. And you'll see that that hour actually is going to fly by because you're not distracted, you're engaged in it. And being active and engaged in your learning is what makes it more memorable. Okay, so this here is Olivia. Olivia, this video was recorded, I forget, less than a month, I think. I forget, I think it was like 4 weeks after she started using the course, and she phoned me to say how much she loved it, and she said she wouldn't mind me recording it. So see what Olivia says.
So much easier and so much more bearable, because I know from speaking to a lot of people at school, they've been like, oh my god, there's so much content and we need to know all the content in detail, so you need to know every single little bit of the spec. So it just makes it easier laying it out. Having it laid out is so much, it's just, honestly, it's almost been like a lifesaver. because my biology AS wasn't great. It's literally changed how I've done biology, and I'm doing a lot better now. I've been through the ultimate guide with my AS biology. I look back on it and I'm like, why did I write that? I look back on all the questions and like, why have I written that? And I'll be like, well, because I didn't have this. I was getting D's and C's, and now I'm up with the A's, B's, occasional A stars and some things. And my teachers at school are going, hang on, how have you? Because they've been like, oh, what have you done this? Like, what work are you doing? That helps the understanding as much as it helps answering the exam questions. Because you can learn stuff without understanding it necessarily, but then this helps with understanding it and then being able to apply this to a question and saying exact, and then hitting the marking points on the question.
Okay, so yeah, her teachers were like, what have you been doing differently? Something's changed. And she'd basically been watching my videos, 3 or 4 videos a night, and then it changed her answer. She's improved her grades massively. So in just a few weeks, it really does work. Okay, so action plan for next steps for you guys. Implement the things I've told you today. Try them out, because if you don't try them, they're not going to help you. You'll see how much of a difference they use. Use fast learning and all the rest of it. And basically, if you can, get the ultimate guide, because I've done all the hard work for you. It's going to teach you through it. Like the ultimate guide, in fact, I'll give you a quick rundown of the 2 different options if you are interested. Then the ultimate guide. Okay, I'm going to start with the revision. Revision course is all the biology videos, and that's basically it. It expires at the end of this year. It's just for the revision videos until the end of exams this year. The ultimate guide includes all of the, obviously, AS and A2 biology videos. They are all in. Oh, you've got it. It's so good. Okay, thanks. Well, you're going to bear with me for just a second. Okay, the ultimate guide also includes all the exam webinar series. So I'm doing a full series of webinars that are going through the AQA Paper 3 essay, loads of questions about that, going through topics 1 to 4, topics 5 to 8. If you've missed the webinars, there's pre-recordings of them. Like, you get more time with me. You get super small groups as well in that. So you get to ask me lots more questions. I deal with everyone's questions. If you have issues on the course, you leave comments on the videos. I get back to you. The ultimate guide includes the exam technique package. There's a whole course on exam technique. That's all included. Whereas the revision course is just the videos. You can't ask me questions, and but it's more self-guided study. But you can obviously use it anytime you want. They both come with the improver grade guarantee. So people have been asking me, if I'm at this grade now, do you guarantee me getting that grade? What we do guarantee is that if you complete the course, obviously you've got to complete the course. If you buy a textbook and never open it, it's not going to help you. But complete the course, and if you have to let us know what grade you're at when you start the course by filling in a form. It's in the course. And if you don't improve by a full grade by the time you've done your exam and you get your results slip in August, then take a photo of the results slip, send it to us, and we give you your money back, so that we know it works. So everyone who went through the course typically improved by 1 to 3 grades, and nearly 2 grade average was the increase for people who completed the course last year. So and you can easily complete it between now and then. So I've got a quick question for you. You, the student, I want to know how much you would delve into your back pocket for the ultimate guide, or for getting access to the videos until the end of your revision, basically, until the course is, until you're free students for the summer. So this is you personally. Your parents may help contribute, or your grandparents. You can break the piggy bank open and see what's in there for a rainy day. You might, some of you, 40% of you have part-time jobs, so it's maybe save up a bit more there. Okay, so you don't have to vote. Okay, but let's have a quick look. So 10 to 20 pounds, so a third of you, so equivalent of a textbook. All the studies we've done said, like, basically, in terms of effectiveness, it smashes a textbook out of the water. So an hour with a tutor, average price for a tutor these days is about £35 an hour. So about a third of you would pay like the equivalent of 1 hour tutoring for unlimited guiding, basically. 2 hours of a tutor, 20% of you, and some of you, like, effectively the decision you've got now is like what grade you need to get into uni, and how much work is it worth doing, and how much work is it worth paying in order to get to the university of your choice. Typically, these are some of the things that people say they struggle with, and the reasons why they don't feel like it's worth paying for the guide. It's too late, and I wish I knew about it before. There's only a month to go to the exams. We get this email every single day. And this is my answer to you. So this is the last week of the full free trial. So these students that you can just about see up here, this is the AS-AQA course, completely full course, exactly the same as you get if you buy the course that people were trialing. Look at this, enrolled for 6 days, 5 days, 6 days, 5 days. Look at India, completed 78% of the course in 2 days. It's about, someone was asking me how many hours is it? It is, I worked it out yesterday, the AS course is just over 10 hours, and the A2 course is a fraction more than that. So 10 hours of video, you can cover that entire first year of your course, entire year in 12 hours. And like, it's pretty phenomenal. It's absolutely like, so the amount of time you need to spend to cover the entire course is minimal. Cost is a big one. I'm about to deal with cost, but like, you've basically done 2 years of studying, most of you, to get to the point where you're at now, and what we're about to offer you comes in at the price of a couple of hours of tutoring. It also saves you heaps of time. Typically, you're going to be able to save that time. You're either going to be able to do more work for your biology, you're going to do more work for your other subjects, maybe you go and do your part-time job a bit more and go and earn some pennies in the same time. So cost is, I can understand, it seems very expensive, but the amount that it actually improves to you, and remember, if you don't improve a grade, you get your money back anyway. So we know it's either going to work and you're going to go up a grade, or you're going to not pay for it. So it looks too good to be true. I'm a bit suspicious. Well, check out the Facebook reviews. Obviously, we don't write them. You're about to write a whole load more of them. I don't know what you're going to say. Also, there's a full satisfaction money-back guarantee. If you buy the course and there's something that you don't like about it, then we give you, just let us know straight away and we give you your money back. Now, if that's after your exams and you've used 3 courses of, and you let me know after your exams that, oh, actually, I didn't like it very much. We had a couple of people try to do that last year. It's a fair use policy. If there's something you don't like about it, let us know straight away and we'll give you your money back. That's straightforward. And how many have I had to do this year? No, I haven't given anyone their money back. Everyone has loved it. I'll just use the free trial. Some people said last week. Well, the free trial is finished for the year. There is a new free trial, but it's a limited version. It's not going to give you access to all the videos. That is gone and done and dusted. Unfortunately, the loophole has closed. And some of you are saying, how long is left? I don't quite know. I don't know what you mean, Jess, with how long is left. But okay, so they are some of the more common concerns. And what we're going to do about cost is basically give you the best possible offer that you're going to get. This is the cheapest. We trialed this last week for a snap time, and we're going to do it again because we think it's worth us doing, and it's definitely good value. The feedback from students was amazing. Like, so what you, the student, are prepared to pay off the revision course. This is biology videos only, remember. The full price is £119. We will match it up to the value of 50 quid. So if you are prepared to pay £50, then we will give you £50 off, which means basically the total cost that you have to split with your parents is 69 quid. That's 2 hours of tutoring. The equivalent of 2 hours of tutoring, and you get me. That doesn't buy you very much time with me. One-to-one, if you want me to teach you the entire course in one-to-one tutoring, you are looking at not getting any change from £3,000 to £4,000. And you've got my best teaching for 69 quid, that comes to. So go and have a chat with your parents. When they see you are prepared to chip some money in and help pay for it yourself because you know how valuable it's going to be, then they will see that you're serious. Go and speak to your grandparents, go and speak to your aunties and your uncles and see if you can gather that remaining £69 and see what you can do. Because, and I said, if you don't improve by a grade, then we give you your money back. And the ultimate guide, to be honest, most people last week chose the ultimate guide just because they wanted the extra time with me in those webinars, going through, giving them peace of mind that they're going to get the best possible prep possible. If they want to ask questions on the tutorials, you can ask questions. If you have email, if you have issues, then I can deal with them basically. You buy a little piece of me when you buy the ultimate guide. So whatever you pay towards the ultimate guide, we double up to the value of £100. So if you personally are prepared to chip in 50 quid, we will give you £100 off. And then your parents or your aunts and your uncles or your grannies or your piggy bank just has to pay the remaining. So that, I can tell you, is like the best, like those, who here has used the ultimate guide? I was going to maybe try a little experiment here. If you're prepared to speak and describe it, I'd be totally rolling the dice. If someone who's used the ultimate guide and paid for it wants to talk to everyone in the room, just, you don't have to turn your video on. If you're willing to do that, let me know in the questions, and I can let a total random who's just You don't have any relatives in there. Lewis said you've paid for it and it's amazing. Lewis, do you wanna say anything in like 1 or 2 sentences to the group? I can probably find you in the list and turn your microphone on and unmute. Do you have a microphone? You need a microphone first of all. Alex Dyer said you will. Are you up for it, Alex? Let me find you in the list. This and Adil. Okay, so let me find you guys in the list. Search. I can search. Okay, I'm gonna go with Alex first. I won't turn your camera. In fact, I don't think I can turn your camera on. Alex Dyer. Are you ready, Alex? Give me a yes in the chat, and I can unmute your microphone. I think this is going to work. So, okay, Alex, you're ready. Okay, 3, 2. This is I'm I'm a little nervous. Don't worry about it. Just give a description. Say what is your advice to people who are thinking about buying it now. I'm going to turn your microphone on. I think. Alex, can you hear me? Yes.
I can hear you. 100% worth it, totally. I used it last year, and I think, like, first off, I finished the course within probably 2 days for my AS, and I think during that tiny period of time, I managed to go up like 3 grades, and I think it was just the really compact, specific knowledge that it gave me. That as soon as I actually did that, within 2 days, like, I started doing past papers just to test how much I'd actually learned, and I found it really helped me to target like the questions and actually start getting marks, because I think before it, I really like knew a lot of the knowledge, but I never knew really how to condense it into a way that was an effective way of getting the marks, because I really hate that about A-level biology, and the fact that sometimes you look at the questions and it's not really, although you might be actually putting the right answer, you're not getting the right marks. But I think the videos really help you to target like how to get the marks and what the examiners really want.
Okay, sweet. Thanks, Alex. That is awesome. So, like you said, 3 grades in, wow. I mean, super effective. And I think I know that your account, you have an Ultimate Guide account, don't you? So that's nice to know that it's definitely working. Adil, I'm going to go one more. Adil, you said you're up for it. Are you still down? I'm going to, again, Adil says, yes, 3, 2, 1. Adil, your microphone is unmuted.
Hi, can you hear me?
I can. You are live, so to speak.
I'd just like to say hi to everyone. The guide is very good. I bought it last Sunday, and I just like shout out to Button. Shout out to Button up.
Good work. Thanks, Adil.
Cheers, man.
So, busting a nut over there by full standards. Okay, so good work, all of you guys. So just basically, if there's anything else that's like why you wouldn't particularly want to buy it, I'd like to know, because all the feedback I've had, basically every day I open my inbox and I see, oh, is it my sound that's not working? I think you can hear me still. Okay, so basically let me know if there are any other objections that I can deal with, then let me know, because I will do that. But I'm going to send you an email with those offers in right now. Everyone's asking me, how do I get to the special offer? Okay, so then I'm going to send you an email in the next 15 minutes, basically, and that email is the only place you can access these offers. So if you want to get the Ultimate Guide as cheap as it has ever been, and it will ever be again, then that is your opportunity. Same for the revision course. Okay, so it's only in 3 weeks. As I said, 2 days you can basically go through the AS course, and probably another 2 days for the A2 course. WJEC, we don't currently do a WJEC course. I'm very sorry, there's just not enough students doing it, and you do too many optional modules. Okay, so how is it best to use it with past papers? Yes, definitely use the course with past papers, or use your own revision. Past papers, try and be doing like maybe 1 or 2 past papers a week at the very least. If you can do 1 a day using the old past papers, then that's awesome. The revision course, it's no good for next year. So if you're an AS student, then basically you need the Ultimate Guide, because otherwise the revision course is just deleted in July. So making notes on every video, yeah, I definitely recommend making notes on every video, just because the process of learning and taking the notes is what's going to help it sort of get into your long-term memory. Jess, you said there's no way. I mean, I'd say if you can't afford it, then you can't afford it. But go and speak to your grandparents. Go and, like, £69 is, yeah, 2 hours of tutoring for over the 2 years. That's basically our tutoring for each year. So it's going to be beyond some, but I mean, that is the cheapest it's ever going to be. So good luck. Can we still watch the selection free? Yeah, there are a few videos in the free trial. Obviously, then use the free trial videos as much as you can. But the full access free trial will not be available anymore. So it's only going to just be for those videos that are there. Basically, loads of you saying thank you very much, super awesome. Yeah, so check your email in the next 10 minutes and I'll get something into you. I'm going to pick a winner, Instagram winner. And also asking you, some of you are asking about using other specifications specimen papers. So if you're an AQA student, is it worth using OCR questions? Normally, I would say no. But this year, because there's so few specimen papers, then I would use some of the old ones. Again, you've got to be confident with your specification. If you know that you do not do the biochemical test for glucose, if you're an EdXL student, then if there's questions on that, you're just going to have to take the other specifications papers with a pinch of salt. But they do have the same maths component. They do have the same stats component. They do have the new style of questions. That's set by Ofqual, who control all the exams. So do use them. Okay, let's pull up my Instagram account. Lots of you dashing in there at the end. Okay, where do I go? Here, my page, back to there. Okay, so where's this morning? Okay, I made a note of the last person who signed up this morning before this webinar. Okay, so basically I'm just going to give it a bit of a scroll in both directions. I'm going to get someone to, okay, so I've just chosen a random screen, basically. You can see random screen. Okay, I'm going to stop there. I'm going to get someone pick a number 1 to 10. How many have I got on my screen? 1 to 9. 8. Both of the first two people said 8, so I'm going to count 8 down. 1, 2, 3, 4, 5, 6, 7, 8. Username is moh.saj1, mosaj1. If that's you, you've just won. Congratulations. I'll send you a message. I need to write that down. So I'll upgrade your account to find that. I might even do the revision tip one now as well. Okay, so let me I actually have to pause my screen so you can't see my inbox, because that would go against my privacy policy. I'm going to go to my inbox. There have been loads of biology revision tips. Okay, so I'm going to count down from the top. I need a number. Ooh, how many have I had? 1 to 62. Lewis came in with 15. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's not 1, 10, 11, 12, 13, 14, 15. Emily Palmer. Congratulations. Emily Palmer. That needs to be written down as well. I will send you an email shortly, but I'm going to send the follow-up email to everyone else. Okay, so let's just go back in here. Basically, Emily Palmer is your gran. It's probably not your gran, that's one I don't think, Lewis. In summary, use the guide if you possibly can. Like, it really has been transforming people's grades. If you don't go up a grade, we give you your money back. I don't know of anyone else in the world that does such things. It's basically what you need. The title of this webinar was How to Revise Facebook Reviews. I'll give you 10 minutes to do a Facebook review. I'll post it on Facebook in 10 minutes. How to revise A-level biology in a week. Use the course if you can, because you can do it in a week. You can do all of ASNA too if you really have to. If you can't use the course, then download the past papers, scan through the past papers, find out what you need to do, write condensed summary notes, do lots of past papers, and practice analyzing the question using the handout I'm going to send you. And Where do you get the link? Okay, I was sending you all of the links. All the replay of the webinar will be emailed to you in 24 hours. The email with all the exam papers that I'm sending you, the links to all the discounts and everything, all the good stuff will be sent to you and in your inbox in the next, well, 10 minutes, as soon as I can do it. I am spent and I am done for the day. So if my computer doesn't want to crash on me anymore, good luck. Again, with the Ultimate Guide, if you've still got biology questions, you can do all the webinars and you can ask me questions and you buy a little piece of me with the Ultimate Guide. So if you have got really burning questions, I'm too busy to do one-to-one tutorials, but I will do as much feedback with you guys as you want. Thanks to all of you. And yeah, it's so nice to hear all your nice things. Bye.